Using bioinformatics to analyze environmental genomics data. Title: Using bioinformatics to analyze environmental genomics data. Introduction. Bioinformatics is a rapidly evolving field that combines biology and computer science to analyze biological data, particularly genome-related information. In this article, we explore how Bioinformatic CS tools and techniques are utilized to analyze environmental genomics data and uncover valuable information about the microbial world. 1. Obtaining environmental genomics data. Environmental genomics data is typically generated using high-throughput sequencing technologies, such as metagenomics and metatranscriptomics. Metagenomics involves sequencing all the genetic material present in a sample, while metatranscriptomics focuses on the transcripts, RNA, produced by the genes in a sample. These techniques enable researchers to examine the complete genetic makeup of microbial communities inhabiting a specific environment. 2. Data preprocessing. Before analyzing environmental genomics data, it undergoes several preprocessing steps to ensure accurate downstream analysis. This includes quality control, adapter trimming, and removal of low-quality reads. Quality control involves filtering out low-quality reads based on various parameters, such as read length, base quality scores, and adapter contamination. These preprocessing steps play a crucial role in enhancing the reliability of downstream analyses. 3. Taxonomic profiling. One of the primary goals of analyzing environmental genomics data is to identify the taxonomic composition of microbial communities. Bioinformatics tools and databases, such as BLAST and the Ribosoma ELB Database Project, RDP, are often used to assign taxonomic labels to sequenced reads. These tools help identify the microorganisms present in a sample and their relative abundances, enabling researchers to understand the ecological dynamics of microbial communities. 4. Functional annotation. Beyond taxonomic profiling, it is equally important to investigate the functional potential of environmental microbiomes. Functional annotation involves linking genes or gene fragments to their biological functions, which provides insights into the metabolic capabilities of microbial communities. Tools like MG-ROST and the Kyoto Encyclopedia of Genes and Genomes, KEG, are commonly employed for functional analysis, aiding in the understanding of microbial processes within an environment. 5. Comparative metagenomics. Comparative metagenomics involves comparing multiple datasets to identify similarities and differences between distinct environments. This approach helps to discern the role of specific environmental factors in shaping microbial communities. By comparing various datasets, researchers can identify shared and unique genes among different environments, potentially providing clues about critical microbial functions and adaptations. 6. Metagenome assembled genomes, MAGs. Metagenome assembled genomes, MAGs, are reconstructed genomes derived from metagenomic data. MAGs provide a higher resolution view of the microbial community by reconstructing individual genomes from the sequenced reads. 7. Network analysis. Network-based approaches are utilized to study microbial interactions within environmental microbiomes. By examining co-occurrence patterns between microbial taxa or genes, network analysis can uncover potential symbiotic or competitive relationships. Tools like CONNET and SpikeEasy are applied to construct and analyze functional and taxonomic networks, offering a holistic understanding of microbial community dynamics. 8. Longitudinal studies and time series analysis. Environmental genomics data can provide valuable information about the temporal dynamics of microbial communities within an environment. Longitudinal studies and time series analysis help tease apart seasonal variations, environmental perturbations, or successional processes. Applying bioinformatics tools like ecology and our packages like DESIC2 allow for the identification of significant changes in microbial community composition and function over time. 9. Machine learning approaches. In recent years, machine learning techniques have been increasingly applied to analyze environmental genomics data. These approaches aim to predict various characteristics of microbial communities, such as taxonomic composition or functional potential, based on the patterns found in the data. Supervised learning algorithms, including random forests and support vector machines, can be trained on labeled datasets to classify and predict the properties of microbial communities. Unsupervised learning algorithms, such as clustering and dimensionality reduction techniques, help explore the inherent structure and patterns within the data. 10. Data integration and multi-omics analysis. Another emerging trend in bioinformatics is the integration of multiple omics datasets, such as genomics, transcriptomics, proteomics, and metabolomics. By combining data from different omics layers, researchers can obtain a more comprehensive understanding of the functional relationships and regulatory networks within microbial communities. This multi-omics approach enables the identification of key genes, pathways, and metabolites that drive specific ecological processes. Visualization tools like MicrobiomeAnalyst and ecology enable researchers to explore complex datasets, identify patterns, and generate interactive visual outputs. These visualizations not only facilitate data exploration and interpretation but also aid in conveying scientific findings to a wider audience. 12. Challenges and future directions. While bioinformatics has significantly advanced our understanding of environmental genomics data, several challenges still exist. The size and complexity of datasets require robust computational INFR, a structure and efficient algorithms to handle the analysis. Additionally, the integration of multi-omics datasets often presents challenges in data integration, standardization, and interpretation. As the field continues to evolve, addressing these challenges and developing novel bioinformatics methods will be crucial. Future directions may include the development of advanced machine learning and deep learning techniques tailored specifically for environmental genomics data analysis. This approach allows for a more detailed examination of the genetic diversity within a microbial community. Single-cell genomics techniques, such as single-cell sequencing and fluorescence-activated cell sorting, FACS, enable researchers to isolate and sequence the genomes of individual microbial cells. 14. Spatial metagenomics. Spatial metagenomics is a relatively new approach that combines genomics with spatial information to study microbial communities within their specific environments. This technique allows researchers to understand the spatial distribution, interactions, and functions of microorganisms within complex ecological systems. Spatial metagenomics relies on tools like fluorescence in situ hybridization, FISH, and shotgun metagenomics to generate data that captures both genetic and spatial information. 15. Data sharing and standards. As the field of environmental genomics continues to expand, the importance of data sharing and standardization becomes paramount. Open data repositories, such as the Sequence Read Archive, SRA, and T.